Welcome to the prototype for Pearson Interactive Labs for Non-Majors Biology. Every lab starts with a real-world story or question to engage non-major students and serve as a hook. You can see here that I'm using my mouse to scroll down, and as I do so, text and images are changing. When we reviewed early versions of this prototype with students, we had a video here for this introduction. And students told us while they liked the cinematic feel, they wanted something that moved at their own pace, which is why we went with a dynamic scroll. After students complete the hook introduction, they're taken to a screen that shows them an overview of the lab experience, starting with an introduction to make sure students have mastered concepts they should have learned in lecture, going into a pre-lab, an experiment, and a post-lab. Students will revisit this screen throughout the lab, and their progress will track to show how much they've completed at each state. The introduction serves to make sure students have retained important concepts from lecture for students who are taking lab in a different semester than they're taking lecture, or serving as a brief refresher for students who may not have mastered concepts. In this, students are being asked to identify in the process of photosynthesis what's coming in and what's going out. Because this is a prototype, not all screens are shown here, and some art is not final. After students complete a brief introduction, they go into the pre-lab. They're asked to build a hypothesis in 2 steps, selecting what they think is going to happen and then why they think it's going to happen with a because statement. While in the introduction, students received wrong answer feedback when they answered questions incorrectly, here there is no right or wrong answer feedback. It's whatever students think is going to happen, so there is no wrong answer, but they will revisit their hypothesis toward the end of the lab experience. After they design their hypothesis, they're asked some questions about experimental design, thinking about what designs could test the hypothesis that they came up with, and then thinking about what variables they would want to use. Then they review some of the steps of the experiment, including the chambers and light levels that will be available to them, and how carbon dioxide moves throughout the system. Then they're asked to think about how to reduce errors in their experiment, and then to set up their graph. Now, in some labs, students will set up their graph in the post-lab, but because the graph is going to plot dynamically as students collect data points in this graph, they're asked to set it up before they begin the experiment. So they label both of the axes and then the minimum and maximum values that they should be using. Then they're asked to make a prediction about what the graph is going to show. Do they think that across different levels of light, carbon dioxide is going to remain the same, decrease, or increase? Again, these have no right or wrong answer. It's just the student's own prediction. Then they launch into the experiment. They click using the plus and minus buttons to set the various levels of light. Again, because this is just a prototype, I'm going to click once and they're going to magically fill in. And then they go into the virtual greenhouse to select the plants they want to use for their experiment. Now here, if I select this smaller plant, I'm going to get reminded of the question I was asked about how to reduce error. And I can try again to select plants going into the experiment that are similarly sized. Now, I click Run, fast forward 24 hours for the student, and then they'll record the various carbon dioxide levels in each chamber. Students will type into these boxes. I'm just going to click once and they're going to populate, and as they type each box in, a plot will appear on the graph below their chart. Now, in the final experiment, students will run this an additional 5 times. And then before they go into the post-lab, they're asked what to do with a data point that seems to be an outlier. Now we go into the post-lab, and students are asked a series of questions, making sure they understand what their data is showing. What does it mean? Then they look at the prediction that they made about what the data was going to show, compare it against the final data, and are asked whether their prediction was supported or not. And then they do the same with their hypothesis. This was the hypothesis they formed in the pre-lab. Here's the data they collected, and they get to decide whether or not their hypothesis was supported. Then, with a couple of final screens, they're done with the lab.